All right, let's get started. Yep.
I just want to understand, is that extra credit for the whole course or?
So there's a question. So there will be an assignment due at the end of this week, unless things go pretty slowly today. Then it might be pushed back to beginning of next week. And I think there's like a couple of questions. So it's sort of a mini project. No, I think this is the concept check. I think both on the mini project and the concept check. On one of them, there's a few points extra credit, and the way it works is and so on the mini project, what that basically means is if the I forget, I think it's out of 30 points, and so if there's 4 points extra credit, then that just means that you potentially could get a 34 out of 30 on that assignment, which for that assignment category, the mini project categories, I forget how much that's weighed, but let's say it was weighed 15%, then you can imagine that any credit accumulated in that category would get weighted by the 15%. So it does potentially allow for extra credit to accumulate in the whole course.
So like the future many projects which will be open-ended, will they even have bonus points?
Well, so I may end up adding a little bit of structure to some, but I don't have specific plans for there to be open-ended, extra credit. But if I see opportunity, I add that. I may occasionally just add those things in when there's something that I think that would be worthwhile for me to incentivize, but not something that I want to subject everyone to. So that's where that comes up. Otherwise, the Muddy's Point assignments, thanks for those. Those are very great. Those are great. I went through everyone's, and on things that I didn't think would be kind of immediately addressed in class over the next week or so, I gave responses to, so you can check your responses to those Muddy's Points to see if there's any comments about that. And I guess that's all I have for announcements. So last time, and let me just get my notes out to keep me honest. So last time we had been talking about introducing the evolutionary algorithm, a form of population-based metaheuristic. And so didn't quite get as far as I was hoping to get. So we're going to backtrack. I'm not going to backtrack a little, but I want to sort of finish up the discussion there and then go into the evolutionary algorithm itself, and then the genetic algorithm and instantiation of an evolutionary algorithm. All right, so last time we talked about these evolutionary algorithms as which are these population-based metaheuristics, optimization metaheuristics. Where the idea here is that you start with some population of candidate solutions. And so you have sort of an initial cloud of candidate solutions. And so each one of these, well, I can say C1, C2, C3, C4, some population level. So there could be maybe N of them. And the idea is each one of them could be evaluated in whatever your optimization objective is. So, so you could imagine you could take each one of those and you could then say, I'm going to evaluate F of CI. So, so this is the actual optimization objective. And you can say, how well do each one of these candidates do? And so you're going to find that some candidates are really terrible. You never want to use those. Some candidates are really great. So you probably, that'd be fine to use those, but you don't know if they're the best. And so the goal then is to then take how well each one of those did and pipe that into your evolutionary algorithm. And this evolutionary algorithm is going to maybe kill some of those and maybe amplify others. And so it will then produce, and I guess I'll do a little dotted line here, a kind of new cloud of candidate solutions. So it may keep some around, like C1 and C2 might be kept around, but C3 might have been killed and C4 might have been killed. And those ones might be replaced with C1 prime and C2 prime, which might be variations of the ones that performed well. So imagine if, I can say, imagine C1 did much better than C2, which did much better than C3, which did much better than C4. And so these C1 and C2 were kept, these C3 and C4 were discarded. And then that new population could then be brought back in to evaluate, to do this all over again. But in any one of these algorithms, there is a termination condition. And if so, then we can return population of solutions. And then otherwise, if we haven't reached the termination condition, we can just restart. And so the idea here is that this termination condition, there are some questions about this in the muddies points. This is a little bit open-ended, but the basic idea here is you might have a budget where you can only evaluate this F a certain number of times. Once you hit that budget, you got to stop the process. Or you might have a convergence condition. So it might be that your population tends to be hanging around a particular solution set and you're not managing to get any diversity in the population. Or it might be that even if you have diversity in the population, the optimization objective is sitting around a particular value and you can't make it, no matter what you do to search the space, you don't ever really get much better than that value. And so if you set a threshold for these things, then you can say, all right, we're not making any progress, we're not making any improvements, we're just going to stop now. And so because these are metaheuristics, they're not exact solutions. We never know when we've reached the optimal solution for sure. So we need this termination condition as a way for us to tell us that when we've reached the kind of good enough state. So, I kind of mentioned in some of the responses to Buddy's points, a metaheuristic is kind of a good enough optimizer to find good enough solutions. So it's like a heuristic to find heuristics. And so this termination condition is what we mean kind of, it's good enough. We don't think we're going to get much better than this on the computational budget that we have. Question?
So let's say when you were choosing your initial candidate solutions, they were just scattered all over, they're not even close to each other.
Mm-hmm.
Is it usually the case where once you run this algorithm a couple of iterations that you'll find that they all kind of converge to the same place, or do you usually find them like in different places and just pick them up?
Yeah, so the question was, you start off with a random group of solutions, candidate solutions that are scattered everywhere. And then what typically happens after this population has been reshaped? Do they end up clustering all in one area? And that is going to depend on the optimization objective and maybe how spread out these things here are. And we'll talk more about this because there actually are sometimes good cases for why we want to design evolutionary algorithms to maybe maintain diversity in the population. So that we can end up getting, and we'll talk about multimodal optimization, for example, when you're not just looking for one peak, you're looking for all the peaks. We'll talk about multi-objective optimization, which gets a little more complicated than that. But so the answer is it kind of depends on the shape of the optimization function, because if the optimization function is multimodal and the peaks are kind of close together in fitness, and I'll define what fitness is here, then you could imagine that you might converge with some in each peak. And so some of your population will be here, and some of your population will be down here. Well, yeah, so these are kind of the in solutions, I'll say the in solutions after EA has run. And this is for a maximization problem. And
Can I
so
ask
they
a
might have,
question?
yeah, I'm sorry, there's a question online. Go ahead.
So if we're asked to implement this algorithm, would you mind if we put the termination condition right after the optimization function? You know, I think it's cleaner that way instead of evolution algorithm happening first and generating new things and then hitting the terminating condition.
So the question online was, can this termination condition also be put up here? And that's fine as well. So yeah, this is for purposes of kind of, saying what the EA was, I kind of put the termination condition here just because I don't want to focus on it but yes, of course it's fine to implement the termination condition up here rather than going through the step of this optimization. There are some cases where the evolutionary algorithm might do a filtering that is beneficial, and so you might actually value the solutions coming out of the EA. But this is very generic, and so we are going to see a genetic algorithm and how it's implemented, and things that get a little bit more concrete. But just generally, yes, it's fine for this termination condition to go up here. I don't really want us to get stuck too much on worrying about the termination condition, because really the magic that we are engineering happens inside this box over here.
Sure, thank you.
Any other questions? Yeah. So the question was, is the termination condition, is there a termination condition per candidate or for the whole population? And it's for the whole population. So it's for the whole algorithm. How long do we want this algorithm to be reshaping this position, this population? All right, so that's kind of the generic evolutionary algorithm. It's called an evolutionary algorithm because it starts with a population, and based on their relative performance, or we'll refer to that as their relative fitness, then you get a new population out that hopefully has a better set of performers, and that population gets further adjusted. And that reshaping of a population so that you change the frequency of certain solutions ends up being what we refer to as evolution. So broadly speaking, the term evolution just means the change in the frequency of strategies in a population. And so we are building an algorithm which tries to create productive changes in the frequencies of candidate solutions. All right, so this it's supposed to look a lot like kind of a cartoon of what we think goes on in biology, in living systems. You have a population, you have say a species that has a bunch of different particular strategies at, some might have a longer neck, some might have a shorter neck, et cetera. And so, well, what's the optimal neck size? And so you could imagine that you send a bunch of these things in, see how well they do with their neck size as it is. Based on how well they do, you basically can think about each one of them having a different number of children. And because there's a finite number of resources, not everybody gets to have all their children. So those who have more children have a greater likelihood of that strategy staying in the population. Those that have fewer children have a lesser likelihood of that strategy persisting. And so over time, then the optimal, in other words, the neck size that produces the most children, is the one that is going to be represented the most in our population set. And so sort of mimicking what goes on in a real biological system. And so with that, let's introduce some terms that are a little more customary to the biological literature that we will then adopt when talking about the genetic algorithm. So this is kind of, I'll just title is kind of toward the genetic algorithm. And so in order to get toward the genetic algorithm, we need to talk a little bit about basically a brief intro to population genetics. And that allows us to talk not only about these evolutionary forces for good, but also this sort of annoying force of drift that we have to deal with. It's really important in algorithms that we build because we work with such small population size. So, all right, so in living systems, so these are key words that are, I'm sure, that are familiar to many of you, but we know that that information about strategies in a living system is encoded in DNA, and you can view this as sort of the code, and that DNA is made up of chromosomes. And if you want, you can think of the chromosomes as sort of different degrees of freedom that we have the ability to work with. They're just sort of different places in which we can package the kind of code of life. And so these chromosomes are, each one of them are strings of genes. Genes being being the operative term here. And we can think of genes, alternatively, we can say genes are locations on chromosomes. So, when people say, do you have the gene for, blue eyes? They're not actually referring to the gene. They're actually referring to a term about to introduce, an allele, which is actually a value that a gene can take. So a gene really is a location. It's where blue eyes are stored. And then you can say, well, what do you have stored at that location? Well, that's the allele. So, so the different options for a gene, I'll just write this over here. Are called alleles. A-L-L-E-L-E-S. And so you might have an allele that gives you blue eyes. You might have an allele that gives you brown eyes. So that's assuming that eye color is stored on a single gene. Eye color is not. But there are other weird things like earwax, whether it's waxy or tacky. That is something that we can talk about as being stored on a gene. So we can say I have the allele for tacky earwax, but or I have the allele for waxy earwax. And so those alleles are sort of together what makes up an individual. So the collection, and if we can call that, so the collection of alleles in an individual is its genotype. So you might refer here to people saying, oh, can we, in bioinformatics, talk about genotyping? Well, what genotyping is actually going down through my DNA and reading out all of the different alleles that I have at all the different locations that those genes are stored. And that gives a blueprint of the recipe that makes up TED. And so, that is my genotype. All right, so now the genotype, how can I say, well, how do we know which genotypes are better than other genotypes? Well, that's going to vary very much depending upon your environment. And so so there is a function which I'll call here, and this is just non-standard notation, but I'm going to call it p, lowercase p, and it's a function that takes a genotype and it takes the environment and it produces a so-called phenotype. So really, when I say blue eyes, I'm kind of jumping ahead. So the genotype is actually the code, and there's a question online, I'll get to that here in just a second. The genotype is really the code, and the phenotype is actually the expression of that code after it's been executed. And the code cannot be executed without a platform. I can have the gene or the allele for blue eyes, but if blue eyes requires a blue pigment from the environment, and I can't find that blue pigment, then even though I have the code for blue eyes, I won't actually have blue eyes. So the phenotype is actually the expression of the allele after it's been kind of combined through the environment. And there's a question online. Let me pick that here. Question on time. Do genotypes always manifest as phenotypes, or only some of them manifest physically? So it's an interesting question of do genotypes always kind of show up? And let me clear that from the other machine here. And the basic idea here is that there is a non-trivial mapping between genotype and phenotype. It's actually, and so we oftentimes don't know, like for example, blue eyes, that involves multiple genes. And so the effect, it's not so simple as to say, I just have the gene for blue eyes. And so you can get these multi-gene interactions, which make it hard to sort of associate one gene with one phenotype. But the system of genes, the whole genotype, really produces the phenotype there. Yeah.
So, like, for example, how would you know if someone is missing the gene, like an allele in their genotype, or just is missing something in the environment, which is why they don't have, I don't know, like purple eyes, for example. Like maybe could someone have the genotype for purple eyes, but there's no pigment in their system for it? Is that why it doesn't show up? Or?
Absolutely. There can definitely be cases where just because you have an allele for something or a set of alleles, then that allele may not actually bubble its way up to the surface and actually express itself in the phenotype, because there may not be there may be a limiting product from the environment. And so just knowing people talk about the predispositions for cancer as another example of that. You might have an allele that may gives you a predisposition for a cancer, but it turns out that it only increases your risk factor a little bit, because really what drives that cancer are things that happen through your developmental history. So exposure to smoke, exposure to radiation, those sorts of things. For someone who has the same smoke and radiation exposure and whatever, you might have more of a risk than they do. But just because you have the allele for that doesn't mean that you're going to get the deleterious phenotype. So the things that are actually, imparting function or risk to you, those are phenotypes. So phenotype is sort of where we measure function. So this is where function is. And the genotype is sort of where it's kind of it stores information about strategy.
What is the letter P stand for?
Yeah, so this, I wrote down here, G by E to P. So when you talk to biologists, then let me just bring up this chat here, and I will answer that question here in a second. So biologists will refer to the G by E to P mapping, and that's genotype by environment mapping to phenotype. So once upon a time, when people had kind of a more naive perspective on genotypes and phenotypes, people talked a lot about the genotype to phenotype mapping. Well, now they don't talk about that anymore. They talk about the G by E to P mapping. So that implies that we really have to understand the environment around an organism to really understand how the phenotype is expressed. And this, and I know there's a question online, I'll get to that in a second. And so this here, we refer to as being, or as expression. So we say that phenotypes are expressed. So do you express this phenotype? You may have the allele for this particular whatever. You may have you may have the gene, the allele, but do you express that gene? And that may depend upon other things in the environment. And by in the environment, an interesting thing is it also can depend on the microenvironment inside your cells. So, and I don't want to get too much into this because this gets where, now there are futuristic evolutionary algorithms that actually start taking on a character like this, but we don't quite know how to use them right yet. But in living systems, you may have heard like of the central dogma from maybe, high school or whatever, where we're actually just talking about COVID vaccines have been kind of a refresher in all this, where you've got DNA gets transcribed into RNA and then gets translated into protein. And this is the so-called central dogma. And there may be some feedbacks as well here. So this is the so-called central dogma. And the interesting thing is these chemical reactions here can be limited themselves. And so the inside an individual cell that has the DNA, it may have a hard time producing the RNA because just the things it needs to build the RNA don't happen to be very plentiful in and around that cell. And then the things the RNA needs to end up generating the protein also aren't that plentiful around the cell. And so what you can end up getting is that inside a single individual in a population of cells, you can get stochastic expression. And so even though you might have the gene for a particular pigment or whatever, if you actually look across your cells, then for a single individual, this might be the nominal expression in the center here, but you end up getting cells, some that express more, some that express less. And so even if, so this environment also includes these small scale features. And that's why in biology, this mapping is actually viewed as a stochastic one. And so generally, we actually can say that there's randomness inside here as well. So I'll write randomness too. Now, what's cool about that in biology is that sets up the ability for organisms to do things like bet hedging. So you can actually have, or even if you had across a family of clones, so if you had 10 animals that were clones of each other, they might all actually express very different, let's say, heights. And you might think that they were expressing different alleles when they actually have all the same allele, but that allele happens to produce a randomness in the developmental pathways for when they actually mature. And some mature with shorter necks and some mature with longer necks. You say, well, why would you want one gene or one allele to produce multiple heights? Well, it might be that you don't know what the environment's going to be like. And so if you can produce multiple heights, you can counteract for randomness that's in the environment, and you can kind of bet hedge. And so a gene can survive for longer if it actually has random expression, because it actually gives it the opportunity to not only do sort of whatever is normally good, but gives it a little bit of variation. So if things get weirdly bad, it can still have some individuals who carry that gene that survive. And so there actually can be reasons why it's beneficial for this to be a random mapping. And in biology takes advantage of this randomness. This is something that the algorithms we're going to build that are mimicking this do not really take advantage of this randomness the same way biological organisms do. But that's something that people are starting to work with. All right, so there was a question a bit ago online that said, so there can be dormant genotypes that never turn into phenotypes, but just remain inside the individual. I think it's probably fair to use that wording, that there can be, this is the big reason why, 23andMe and these companies can end up profiting, because you don't know necessarily what all these alleles are. You don't know what's sort of just sitting there, but you can have a company genotype you and actually tell you all of your alleles, or all of the alleles at particular locations of interest, some junk stuff that they may not know, or just don't bother reporting. But you don't, they haven't kind of expressed themselves yet. They haven't been turned on. So the expression of a gene is going to depend upon triggers from the environment. And so it's possible that a gene can just sit there, just like a subroutine in a program that's never called. So that's, so yeah, definitely these things happen. Yeah.
I had a question on the algorithm that you showed earlier. When you're generating like the C1 prime and C2 prime, I'm assuming those are just small perturbations away from the original C1 and C2, right?
They can be small. I mean, conceptually they're usually, but they could be large as well. And we'll talk about, that's what we're getting to. The GA will actually give us a recipe for how to make these perturbations.
Okay, that was my question.
Yep. Any other questions about this general? The big thing I want you to pick up here is terms like genes, chromosomes, phenotype, alleles. So these are things that we'll see come up again when we look at genetic algorithms, because the developers of genetic algorithms have borrowed some of this language. So we're going to want to know where it comes from. You see if there's any that I am missing out on. Okay. All right, so only other minor terms that I'll mention up here. So I've mentioned, phenotype. Now, phenotype is sort of everything that you express kind of all at once. But we can break phenotypes down into characters. And so we can say a character is a component of a phenotype, and a trait is a possible state of a character. And this is really where the eye color comes into play. So you could say that the character is eye color, and the traits are blue, brown, etc. So just like there are multiple alleles that can live in genes, you can kind of think of there's multiple traits that a single character can take on, and the suite of characters together are the phenotype. And initially, when we were talking about evolutionary algorithms, we're going to talk with relatively simple optimization problems where we're really only focusing on one character. So we're really saying we want to optimize the speed of a process. We want to optimize the cost, et cetera, et cetera. We're focusing on one thing at a time. Now in biology, multiple characters matter in different ways. So a particular character that makes you attractive to the opposite sex might be a character that also makes you very conspicuous to a predator. And so there you've got one characters involved in kind of two different optimization pressures. Or on the flip side of that, you might have one character that gives you an advantage, maybe running from a predator. A totally different character gives you, is not related to predation, is related to the efficiency of how well you sleep or something like that. And these two characters may seem like they're independent, but in reality, at the genotype level, they're coupled together through several genes. And so it's not possible to be better in one character without being worse in another. And so fitness ends up being a superposition, and that's something else I have to define here in a second. How you perform in your environment ends up being a superposition of how you perform along each one of these character axes. And that's where we're going to move on to when we hit multi-objective genetic algorithms, actually bringing on the full force of being evaluated, not just by one thing, but by many things all at once. All right, so then that, I use the term fitness. And so fitness basically is a function of the phenotype and all of its characters. So this is all of its characters. And the fitness can be defined as basically the number of offspring associated with an individual and their phenotype, or associated with the phenotype, really. So a lot of times we hear the word fitness bandied about, and it's difficult to sort of figure out exactly how to operationalize it. In population genetics, fitness is simple. It's just the number of offspring produced. And in the genetic algorithm, it's just the number of offspring produced. Now, what we'll see that's important is the related term of relative fitness, which basically is going to be the fitness of an individual phenotype basically divided by the total growth in a population. And so, in other words, how well are you doing relative to how well everyone else is doing? So you can think of it as the fitness of an individual divided by the average fitness. And so there are a bunch of measures we can have that represent the relative performance of an individual. So everyone is having offspring. I mean, some might have zero offspring, but in general, every phenotype is having offspring. So every phenotype has fitness. So in some ways, every phenotype is fit. But in some ways, we don't really care exactly that they're fit, but we care how fit they are relative to how fit everything else is. And so that's this idea of relative fitness. And this relative fitness will be the tool that allows us to decide which individuals to keep in a population and which individuals to discard and replace with variations of existing individuals in a population. Okay, so it's fitness and relative fitness. All right, so if I put all of that together, so any questions about that? So I've put all of that together, then in biology, we basically start with a genotype. And that genotype is going to produce or express a phenotype or a set of characters. And then that phenotype is going to produce a fitness or relative fitness. And then that fitness is going to get pushed through a possible limitation which is going to change the frequency of alleles in the population. So that's going to lead to a change in frequency distribution of alleles. And this possible limitation is often what is related to something we're going to call selective pressure. So selective pressure is the disadvantage that a phenotype feels. And so you say that if you have pressure against a phenotype, then that means for a particular environment, then that phenotype is going to do less well than other phenotypes. Now, if it wasn't for this resource limitation here, that would be fine. Everybody would just keep producing offspring. But in the end, there's going to be a finite number of resources. There's only a certain amount of food out there or whatever. And so those who produce very few offspring, those offspring probably aren't going to survive. They aren't likely to survive. And so that's what gives you this loop, which makes it so that only the fittest individuals manage to survive. Question? Well, so the model-free discussion is a larger discussion about metaheuristics in general. The genetic algorithm, which will be based off of this sort of approach here, doesn't require any additional information about the shape of the optimization function. It just requires you to say, I have a candidate solution, I can evaluate it in the function. An approach that makes use of model information might say, I know gradient information. So when I evaluate my candidate function, not only can I see how well is it doing, but I also can see what's the slope at the exact point. So I can say, if I were to modify it in this particular way, then I would move up or down. And so if I don't, these metaheuristics, you don't provide them any other information about the optimization objective. You just give them the ability to evaluate the optimization objective at a point, which makes them sort of model free. Does that make sense? I see. So the question is that the function that maps genotype to phenotype takes into account the environment. And so, so then the question is, do we have information about the environment? And the assumption here is that when you put your candidate solution into the optimization objective, that it's a black box. And there may be environmental parameters that you just don't have access to, but it has access to. And so it could be a physical system. And it might be that if you run your genetic algorithm and it's cold outside, you'll actually get different results than when it's hot outside. And so if you're implementing this genetic algorithm in a physical evolutionary loop, then you probably need to control for temperature, because so in that case, you do kind of have control over those environmental variables, but it's at a physical level. Does that answer your question? I thought I heard somebody unmute. Is there a question online? No, okay. All right, so this is the basic loop. We'll call this the natural selection loop. And natural selection is one of the forces of evolution. So this idea is that fitness is driving changes in the proportion of different candidates in the environment. That is so-called natural selection. So fitness is causing certain individuals to be selected to have higher proportions in future generations. The thing is, and that provides an optimization force that we will try to leverage when we build our own genetic algorithms. So that's great. But the thing is, in any one of these systems, natural selection is not the only force acting on these populations. There are parasitic forces that just come from using an evolutionary algorithm or using a population-based approach that we have to deal with. And one of those is drift. So I'm introducing this term, drift, or genetic drift. Sometimes maybe called allelic drift. And so, so if we think about this loop that I just drew, but let's imagine I'm in a population where there's no fitness differences. So I've got my genotype, it produces a phenotype. That phenotype, let's say that all equal fitness, which basically means all produce same number of offspring. Well, then if I go through a limitation step and then loop back around the same way I did before, then I have to ask, what's going to happen to the population over time? So what do we mean by this limitation step? We can show that, we can depict that as saying that in the previous generation, let's say I had a bunch of alleles for strategy A, and I had another bunch of alleles for strategy B. So I had 5 of A and 5 of B. And they're all going to produce offspring. And let's say they all produce 2 offspring. So in that case, my population is going to have 10 of A and we'll have 10 of B. but we know that the population's resources can't accommodate 20 individuals. What does this mean in an algorithm? When we actually write up the genetic algorithm, it's become more concrete. But for you, it means that you only have enough memory to keep track of 10 individuals at a time. So there might be a transient step where you create 20 individuals. So a parent and one offspring, or maybe all the parents die and you have 2 offspring. That's another way to view this. But because your algorithm is only supposed to keep track of 10 solutions at a time, you have to limit back down to 10. And then we have to ask, all right, we're going to get 10 individuals here, but if we just draw them randomly, then what is the population going to look like here? Well, nominally, on average, it's going to have 5 of A and 5 of B, because I've got half A, half B in the middle step. So I'm going to get half A, half B in this step. But that's on average. Realistically, just by random chance, by dumb luck, you're going to get an excess of one over the other. So by dumb luck, you might get, for example, 6A and 4B, just by luck. Because if you think about it, it is actually much more likely for us to get an excess of one than the other than to get exactly the same amount if we just draw randomly out of this group. And then so from there, then if this loops back around, if these A and B still have equal fitness, what's going to end up happening is I will have slightly more A than B here. When I double the offspring, I'll have slightly more A than B, but actually even more A than B over here. So but the proportion, so here I've gone from a 60-40 proportion. This will be a 60-40 proportion, but with slightly more individuals. And then if I limit back down to 10, then it's even more likely that I'm going to have a greater excess in A. So it's very unlikely that I'm going to go back to 5A and 5B now that I have an excess of A. It's even more likely that I might go to 7A and 3B. And this thing will keep going on and on and on until I reach a state of fixation, or where the population becomes fixed at 10A. And so once I have a population that is all one of this, all one allele, then this process can't change it anymore because there's no diversity left to make a change. And so it's going to appear like A beat B. But in reality, there was no fitness difference between A and B. And so our conclusion that A is better than B is not really a good one. Now, you say, well, that's not a big deal because A and B are equally fit. They're just equivalent solutions. It's a lack of convexity in the optimization function, if you want to use that kind of language. But this cartoon example is in the special case of all equal fitness, but it still applies when there's small fitness differences. So it might be that B was slightly better than A, but the amount that B was better than A was not great enough to deal with this random sampling effect. This, at small population sizes, this fixation on a random allele happens really, really easily. And it's also guaranteed, by the way. So natural selection will eventually reduce differences in fitness. And then we're always going to land in this drift box once the fitness is. So this really happens whenever fitness differences are low. Then you're in this situation. So we will always end up fixing your population if you don't have this other thing that we haven't talked about yet called mutation. So without mutation, you're guaranteed that any evolutionary algorithm is going to lead to a population that has all one allele. And that's because the fitness differences will end up becoming so small that they'll be taken over by the drift force. And then drift will cause the situation, it basically becomes if you're familiar with modeling with Markov processes, this becomes a Markov chain with an absorbing state, with actually in this case 2 absorbing states, all A and all B. And if you happen to be closer to 1, then and this is an unstable equilibrium in the middle where they're the equal cases. And so this will end up perturbing itself going towards one or the other, and then you rapidly approach that absorbing state of all A or all B, and then you stay there forever. Yeah.
What about the part where you actually evaluate your A or B, and like let's say you were drifting towards A in this case when you said B could have been slightly better. When you evaluate them in the cycle, when we're doing that like diagram that you were drawing first, wouldn't we be able to, or wouldn't the algorithm be able to see that B is actually better and be able to correct itself?
So the question was, shouldn't there be a way for us to detect when drift is happening? And when drift is going against selection, then is there a way we could sort of stop it? And the issue here is that these algorithms kind of do not have a bird's eye view of what's going on. And so we're kind of trusting an algorithm back here in the selection step. The algorithm is evaluating the fitness of each individual. I guess I don't really want to go about that far, but I'm evaluating the fitness of each individual solution and competing them against each other, knowing that very often there's going to be small differences in fitness. And there is advantages of using random selection here that is proportionate, but the disadvantages, and so that allows, I can get rid of some of this by using, say, a deterministic limitation step. So I could say, I want to match the proportions exactly. Whatever they were here, I want to match them exactly here. But there still could be quantization error. So it might be that the proportions here, when I have 20 individuals, when I divide by 10, I would end up being left with half an individual. And so I end up still increasing my proportion slightly of one over the other. So there are a bunch of these weird random sampling approaches that just happen when you push things down through this kind of bottleneck here, and that we have to kind of deal with. And all of those types of approaches where you get a change in alleles for reasons unrelated to fitness, we are referring to here as drift or genetic drift. And this is really bad at low population. And so this is particularly bad at small population sizes. And so there is a guy who was hired here at Arizona State University a couple of years ago named Michael Lynch. And this is some of them people call him like the new Darwin. Like he has extremely prolific work. He moves into an area of biology. He completely revolutionizes it, sets it up in a totally new direction, and then moves on and goes off in other new areas of biology. His current focus is on better understanding drift and the macroscopic effects of drift on populations. And so humans, for example, evolved with relatively small population sizes relative to, say, bacteria. And he can look at things like, and again, we haven't talked about mutation rate, but mutation rates and energetic loads and things like that across bacteria and across giant vertebrates like humans. And they all sort of are consistent with this idea that drift is very, very apparently influential in the history of humans, where we evolved in small populations, and less influential in the history of bacteria, which evolved in very large populations. And so this is one way to explain why you can get the persistence of phenotypes or traits in a phenotype that appear to not have adaptive value. And it's because the adaptive value they provide over other traits might have been significant, but the population size was so small that the drift forces didn't allow selection to act. And this is really important because when you set up genetic algorithms, it's going to be tempting to always build small populations, because if you build gigantic populations in your genetic algorithm, it's going to take forever to evaluate those 1 million candidates every iteration through. So we naturally like to have small population sizes, but these small population sizes are going to have these drift issues. And so that's why we need to kind of keep those in mind. All right, so that's kind of the basics of population genetics before we can get into the specifics of the GA. Are there questions about this just sort of general framework? Yeah.
Is there a way to like quantify, population size versus the amount of genetic drift you have, or is it just kind of like a general guideline?
Great question. So the question was, can we quantify this force of drift and how it relates to a population size? And yes, this is possible to quantify, but that really is, more work has been done on that. So people don't do that with evolutionary algorithms as explicitly as they do in population genetics over in biology. And so there are methods for that, and they're gradually being imported from biology into the evolutionary algorithms literature, but the richest literature is over in biology. So if you're interested in that, you should look up things about population genetics, about drift, and the so-called drift barrier. And so the drift barrier is a term actually that Mike Lynch came up with when I think it was at Indiana. And the drift barrier is sort of a way in which you can say, for a population size, what's the kind of minimal effect that selection can generate, and beneath that effect, selection becomes kind of, it can't go over the drift barrier. So it sort of sets the threshold for how big of an effect selection has to produce in order to not just get washed away by drift. So that's something that definitely there are people doing that. Not as much. I think it's definitely possible to do a lot more of it with the EA, but people just kind of aren't thinking that about that way. And there are lots of reasons for that. We can talk about that as we kind of keep moving forward. But any other questions about these ideas between selection and drift and how these are two opposing forces? Yeah. Great question. So that kind of allows me to pivot to my next slide here. So the if we think about the effectiveness of LAs and or VAs, and this is kind of a graph of my own. So if we think the y-axis is the effectiveness of an evolutionary algorithm, and the x-axis is the so-called selective pressure. And the selective pressure is, we get the most selective pressure when we have the highest phenotypic diversity. So we have high phenotypic diversity. Or I guess I could say, another way to say that is we have high fitness diversity. So high fitness diversity. Now, generally, one way you can get high fitness diversity is, or at least a heuristic for getting high fitness diversity, is to have high population diversity. Now, this isn't always the case, but generally, if you have a bunch of diverse genotypes in a population, you will likely have a bunch of diverse phenotypes in a population, and you will likely have a bunch of differences in fitness. And that will give you really high selective pressure, because the ones that have really high fitness are going to have tons of offspring relative to the ones that have really low fitness. And gradually the population, well, not gradually, but very fast, the population will get pulled in that way. So if you want high selective pressure, just initializing your population to have high genotypic diversity helps guarantee that drift is not going to be, is not going to start out as something that is playing a dominant role. But the thing is that as you select for solutions that are doing much better than other solutions, you're going to squeeze the fitness diversity down, and the genetic diversity will also be squeezed down as that allele ends up becoming more and more dominant. And so there is a natural progression of things where you might start, hopefully, over here. So this is where you start your algorithm. But then from the start, there's a natural trajectory that kind of looks like this, that moves in this direction. So your EA actually gets better and better, and then gets worse and worse. Now, what do I mean by, why would it initially be really bad? Well, so, and then over here, this is where you have low population diversity, or low genetic diversity. Well, yes, right. So you could think of it in this case, on the far end here, gradually you've fixed. So this is where you reach. This is fixation. So you've had one allele that has become just spread over the entire population because its fitness, the differences between its fitness and all the other fitnesses weren't enough to dominate over drift. Drift is a Markov chain with an absorbing state where one population takes over, and there you go, fixation, it takes over right there. So you always end up with fixation here. Now, initially though, my claim is that intermediate diversity, this is sort of the best case scenario, is that this is sort of the best performance of the EA, not when you have maximum diversity. Because if you think about it, when you have high population diversity, you are extremely sensitive to your objective function, to the point where it is really easy to have rapid shifts to local optima. So if I have an optimization objective F, and that has these like peaks, all these different peaks here. And let's say that I want to minimize. So I'm trying to minimize F. Well, ideally, I would like to find this peak down here. That is the one that it is the global minimum here. But if I happen to start with individuals all over this peak here, I actually have pretty high diversity in my objective space here. So this will have relative different, so the fitness differences will be pretty high here. And what's going to happen is that the ones up here are going to be quickly discarded. The ones up here are going to be greatly rewarded. And I am going to get a super rapid shift down into this local suboptimum, and I'll have no way to get out of it. So when you have really high selective pressure, you tend to get stuck in local traps. And so in this region over here, you get stuck in traps. But then in the other region, in the drift dominated region, in that case, I can get what's arguably worse than that, where I have an optimization objective, and let's say there's not much difference in fitness. I would like to find this, but if I have a bunch of individuals that are close in fitness, then a random one will get picked and it won't even be on an optima. Like it's not on an inflection point. It's a totally random solution. So in this case, I can get totally random outputs. And so in the middle, that's kind of the ideal place where you're following gradients effectively. You're discovering gradients and following them in your optimization objective, but you're not doing them so fast that you're wiping out all the diversity in your population before you find something good. And so ideally, you like to be, this is sort of the, I'll choose a different color for this. The ideal spot is right in this middle area because you don't move too fast, but you also still have selective pressure. So it's gentle selective pressure. Question.
So both of these cases that you drew, the two graphs obviously they both have different problems with them, but it seems like if you would just choose X such that X was very varied, not the fitness itself, then you would be good to go. Like in both cases, like because I noticed like on the red graph, for example, although they had large differences in their fitness, but they were still pretty close to each other in X. So if you just vary the X out, then that would solve both of those issues.
Well, the observation was that if you start with a diverse X, that maybe you end up naturally finding yourself in the middle here, because a diverse X will hopefully have, you won't be stuck in this situation where they're all very close together. And the issue is that even if, so I kind of drew this as a cartoon case just to illustrate the point, but even if I had a really diverse set of X, if I happen to be in a case where some of the X happen to be over here, some are over here, it still could be the case that I have a huge difference in fitness, even though the X's aren't close together, that will still cause them prematurely to explore like I can randomly, if I just randomly kind of scattershot over this entire domain, I might just randomly get a huge diversity in fitness. And after the first generation of the evolutionary algorithm, the ones that were lucky enough to happen to find a relatively high fitness peak may attract the entire population. And so, but it might just be that I just haven't happened to find the peak that was over here. Like I only had 10 individuals. And so I don't want to just assume that the 10 individuals I started with have the best solution. I want to give them opportunity to move around this thing. And so that's really what I'm sort of saying. In high selective pressure, this means that in like one generation, you're going to suck away all your diversity on whoever happens to be best in that first generation. I want to keep the diversity around for long enough that I actually explore more of the space to find those peaks that I wasn't even near when I randomly scattershotted in my initial population. But I need to be careful because eventually, I know this fitness is going to be going away. And so eventually I get to this drift region. And so what we're going to learn about generically in evolutionary algorithms, I've mentioned there are hyperparameters to metaheuristics. And so hyperheuristics can optimize the parameters to your metaheuristics. Some of those hyperparameters are things like, so in the ones we've talked about so far, like in the population size. Now when we get into the genetic algorithm, we'll talk about other hyperparameters like mutation rate and crossover frequency and things like that, that are other things that the reason we have those hyperparameters is we are trying to put our metaheuristic in this middle region for as long as possible. And you can crank up the mutation rate and maybe that'll do it. Or you can increase the population size and maybe that'll do it, and so on and so forth. Now, the reason we have hyperheuristics, which I'm not going to talk about the design of hyperheuristics, but you can imagine that the best choice of hyperparameters, the parameters of the metaheuristic, those choices are going to vary with the optimization objective. And so for some optimization objectives, you might need a different type of evolutionary algorithm than for other optimization objectives. Some of your optimization objectives are going to look like this. Some of your optimization objectives are going to look like this. We may not know which bin we're in because we don't have a way to plot our optimization objective. You often don't know what the loss function over your neural network weights look like when you're optimizing a neural network. But if you tried to plot it out, sometimes it looks like an ugly thing that looks like this. So a hyperheuristic will go in and monitor your metaheuristic and try to figure out if it's getting stuck in traps or if it's drifting away. And if it is, it can then adapt these hyperparameters dynamically to try to recover from these things, kind of like the suggestion was earlier about couldn't we sort of try to catch drift before it happens? So, so that's kind of what you can kind of keep in mind is that the goal for either if you're manually choosing these parameters or if you're giving a hyperheuristic to choose these parameters for you, the goal is to balance selective pressure with drift, because alone, selective pressure finds too many traps, drift finds random solutions. And we want them to not converging on a trap early. We want it to search a space for a long time and then gradually converge on a spot that's pretty good globally. All right, so there are general questions about this. Let me make sure there's not chat messages that have popped up too.
Can I clarify something, Professor?
Absolutely, there's a question on online.
For the selective pressure, if it's too high, that's bad because you basically take out any diversity that you have to begin with and fall in the traps of like suboptima and not getting the actual optimal solution. And then for the lower side of lower selective pressure, that just goes into genetic drift, correct?
Right, so with high diversity, you end up basically throwing, you put all of you have high diversity means high selective pressure, and that's bad because of the traps. Low diversity means you basically turn off selection because selection can't act, drift will be the only thing that acts, and that ends up just getting a random solution out. So yeah, those are the two issues.
Got it, thank you.
And there's a couple other little things online, and then I will let you guys go here in just a minute. Is there a similar balance? So there's a question online. Is this similar to the balance between bias and variance? And I definitely think for those of you who've taken machine learning courses, we've talked about the bias variance trade-offs, that you probably could make some nice connections here. I'm gonna leave that to you as an exercise, but I definitely think that's a good thread to tug on, the bias variance trade-off there. Is there a way to measure distance between points in n-dimensional space to figure out how close the points are together, even though they're diverse? Absolutely, we will talk about various metrics. You can basically think of two norms and things like that, and we will talk about how we measure diversity and what we do about that. And then there's another question, is this similar to exploration and exploitation and active learning? And I actually will give you a graph, if not next time, the lecture after that, which makes the connection between exploitation and exploration more explicit, because there is definitely a connection to exploration and exploitation here. So that's all I've got here. So we will introduce the genetic algorithm formally on Thursday, and see you then.
The concept check that's due, like I think on the 21st, is today's lecture and Thursday's lecture is going to be what we need to do that for.
Yeah, well, I will review the concept check to see. It's been a while since I wrote that concept check. I think most of it you'll be able to do from today's lecture, but if it looks like we need Thursday's lecture, I might bump it to make it due next week. All right. If you have anything else, feel free to come up after class. Otherwise, I'll see you Thursday. So if there's any other questions online, otherwise that's all I've got. So I can see you all next week. Or not saying next week, on Thursday. And if nothing comes up, I'll probably just be ending the meeting here in the next few seconds. Okay.